Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Yijun Huang from WuXi Advanced Therapies.
Thank you very much. Thanks everyone here for your interest. Today, I would like to share with you what we have learned so far about Analytical Ultracentrifugation, AUC, at WuXi Advanced Therapies, and where we are now for testing adeno-associated virus AAV-based gene therapies. First of all, a little bit introduction about what we do as an organization. WuXi Advanced Therapies and Oxygen work together to provide end-to-end solution of cell and gene therapy as an integrated contract testing, development, manufacturing organization. Oxygen supports upstream and discovery work, while WuXi Advanced Therapies are able to support large-scale manufacturing for both clinical and commercial. As a whole, we have 8 global sites supporting a large number of programs worldwide. Looking at the map, Oxygen is based in Europe, focusing on preclinical manufacturing technologies. In US, Philadelphia site focusing on cell and gene therapy GMP manufacturing and testing. In China, we have plasmid manufacturing as well as viral vector and cell therapy services. Today, we're talking about how to address one of the critical quality attributes of AAV using AUC. First, I will explain why you should consider AUC for testing AAV gene therapies. Second, I will show you how it works and what information you can get from AUC analysis. Finally, I will demonstrate how good is the performance of this assay. Is it a research tool or is it robust enough for GMP operations? So far, there are at least 3 AAV-based gene therapies approved, and there are many more in clinical trials. AAV is certainly proven as an established platform for gene delivery. AAV can target different disease areas and can be used either locally or systematically. In reality, AAV products are complex and heterogeneous in nature, which are often related to the manufacturing process. Those brings up unique analytical challenges which we have not seen. The critical quality attributes of AAV itself includes viral titer, such as particle titer, genome copy, infectious titer, transduction unit, content ratio, which we are focusing today, as well as aggregation. Traditionally, the content analysis can be characterized by microscope, such as transmission electron microscope. As you can see from this picture, AAV capsids are a mixture of empty capsids with fully filled, and a partially filled with genome containing content. So why it matters? The content analysis is critical because of 1. Safety concerns due to immune response of empty capsids. 2. Impact on gene transfer efficiency. 3. Dose consistency. 4. Lot-to-lot variability. I'm going to show you that AUC is a platform and a robust method for AAV content analysis to determine the capsids ratio or size distribution. Compared with other techniques, AUC certainly provides unique advantages. Greater resolution of empty and full capsids, both qualitative and quantitative. It's performed under native condition. It provides absolute measurement. It's serotype independent. It's reagent free. And the method is also reproducible and relatively fast. For your information, the sedimentation coefficient of typical AAV capsid species is listed here. We established the platform using the new optimal AUC from Beckman Coulter. The workflow basically has three phases: data acquisition, data analysis, and reporting. We have been testing AAV in past 4 or 5 years. Now we have the assay set up in both GMP and R&D settings. First of all, AUC provides a unique tool to support process development. In this study, we were able to characterize AAV content in different matrices from the process. The results generated from AUC can help process optimization, particularly on downstream purification. Second of all, AUC provides robust data to support both in-process testing and final product testing. In this case, we can use AUC to test in-process samples. As you can see, the ratio of full capsid is significantly increased after IEX for full capsid enrichment compared with the sample before the process. For final product testing, AUC assay provides sufficient resolution to distinguish between empty, full capsids, and anything in between, as well as impurities, aggregates. You can get a size distribution graph as a visual presentation and also the list of peaks in a table with sedimentation units and the relative percentage of each peak. It provides both quantitative and qualitative information. I also want to show you that our AUC platform is optimized for GMP operations. We performed an in-house qualification study using a representative AAV sample to evaluate the performance of this assay. In total, we have 5 runs performed by 2 different operators on different days. It is a protocol-driven exercise, and we evaluate key parameters according to the guidance. In this slide, you can see the data we used for intermediate precision. We have very tight ISD from 9 data points across 3 runs from 2 operators. Both the full peak and empty peak has ISD as low as 5%, which is considerably great. We also evaluated the linearity of this test article. In this table, we determined the peak area of the full peak under 3 different concentrations. The plot showed a good linearity. Due to the material availability, this assessment is limited, so the actual range would be wider. In addition, AUC can also be used for stability or formulation study of AAV products. For example, the profile of AAV capsid is very helpful in this case to determine the stability of an AAV sample under a stress test. Both the ratio and the peak area could be indicating the stability. In summary, we talk about that WuXi Advanced Therapy offers an end-to-end solution for cell and gene therapy. We have established the AUC platform and qualified for GMP use to support robust lot release, characterization for in-process and final product. We are also able to support qualification validation for client-specific test article and customer study. Finally, we believe that with a fully integrated approach, WuXi Advanced Therapy can accelerate time to market. Last but not least, thank you for your time.
Okay, so before we get to any questions, we have some questions for you that will help our speaker. So the first question on your screen is, are you currently developing advanced therapy pipeline? Yes or no? So you can use your mouse and go ahead and select the correct answer for you. The second question is, are you currently developing AAV? Yes or no? And the last question is, how soon are you looking to speak to a CTDMO to support your needs? 0 to 6 months, 6 to 12 months, or 12 plus months? So if you can go ahead and answer those questions, our speaker will be grateful. And now we're going to go along to answer your questions. Great. So the first question is, what is the sample requirement for AUC testing?
The sample requirement is different from each sample type. It could also be different from R&D and GMP, so please contact us.
And what information will be reported by the AUC analysis?
So you can get a size distribution graph as visual presentation in the report, and also the list of peaks in the table with segmentation units and the relative percentage of each peak.
And what other parameters are evaluated by the assay qualification?
Please contact us. We can provide a summary report of the in-house qualification. We are also able to perform test article or client-specific qualification studies.
Okay. Well, looks like that's all the questions we have for now. If we didn't get to your question, we will be passing those on to Yijun. You can follow up with you directly. You still have time to go ahead and type in your questions, and we'll get those passed along. Thank you for joining us today. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.